Bacteria produce a wide range of different molecules. Many of these molecules are not required for survival, but give the producer a fitness advantage in their environment. These molecules are called secondary metabolites or natural products. We know from biology class that all organisms have DNA, which is our genetic code. DNA is transcribed into messenger RNA, which is in turn translated into an amino acid sequence. That sequence goes on to form the proteins and enzymes that perform millions of different functions in the organism. What's cool about bacteria is that the genes that encode for the biosynthesis of secondary metabolites are often clustered together in the genome. Bacteria are also capable of transferring genetic material between individuals without reproducing in a process called horizontal gene transfer.
Bacteria have evolved to produce certain secondary metabolites for several reasons, including fighting off competitors, attracting symbionts, and scavenging nutrients from the environment. Several examples of distantly related bacteria producing the same molecule by different biosynthetic mechanisms have been identified. The selective pressures that cause the bacteria to produce these molecules tells the story of how the common ancestor evolved.
So if biosynthetic gene clusters are DNA, and DNA codes for protein, then biosynthetic gene clusters must encode for those enzymes which catalyze the chemical transformations that produce secondary metabolites. These disparate bacteria have different clusters of enzymes. Since producing secondary metabolites requires resources and energy, some bacteria may have evolved more efficient mechanisms for producing the same metabolite. An example would be a more efficient enzyme that can skip biosynthetic steps. Another example could be shunting intermediates from a primary metabolism to a secondary metabolite pathway.
Now let's talk about why this research is exciting. Many bacterial secondary metabolites are bioactive, meaning they have biological activity, and there are many drugs that are derived from natural products. Understanding these biosynthetic mechanisms may lead to the production of new drugs. Molecular biology techniques allow us to modify the genomes of bacteria, including biosynthetic gene clusters. By modifying the biosynthetic genes and the resulting enzymes, we can create new natural products which can be used as drugs to treat all kinds of diseases. Better understanding of how enzymes work will allow us to use them in chemical synthesis for pharmaceuticals, materials, cosmetics, foods, manufacturing, and beyond.